Welcome to SciMind. This video demonstrates how our platform drives pharmaceutical innovation. Let's explore. Scene 1. Identification of structure and activity data in small molecule literature. Upload a demo document, and researchers just need to wait a moment to see the summary of the document. The assistant will also provide the count of molecules and tables in the document. Preview the document and click on the viewer window to see all the molecules parsed from this document. The SciMind platform supports automatic table extraction. We can switch to table view mode to see all the extracted tables. Users can select tables of interest and send them to the dialog area, where the assistant will analyze them. If users want to know which molecule in the table has the highest activity, they can ask the assistant. Extract the molecule with the best activity from the table. The assistant will identify the table information and return the results based on the user's query. Scene 2. Extracting table information from peptide patents. After uploading the sample document Demo 2, the assistant will return the relevant summary and display the number of molecules and tables contained in the document. Then, open the document preview interface and locate the table you want to parse based on the page number. Open Viewer window. Users can quickly match elements in the view by clicking on the table border. Then send it to the assistant to swiftly and accurately retrieve the information in the table. If users want to assess the difficulty of synthesis based on the length of each sequence, they can ask the assistant, which sequence in the table contains the most amino acids? Our assistant will then analyze the table data further based on the information available to provide an answer. Scene 3. Markush structure analysis and scope judgment in patents. In this example, we upload a pharmaceutical patent. In addition to reading the patent content and identifying molecules and table information, the assistant can also determine the core structure in the patent and further analyze the chemical space protected by the patent. To avoid the risk of infringing with a specific molecule, ask the assistant, "Is this molecule protected under this patent?" The assistant will assess based on its knowledge of the patent's chemical space. Here we attempt to judge whether the following two molecules are within the patent's protection scope. We can see that the first molecule is very similar to the core structure, and after further assessment, it is determined that this molecule must be protected. However, another molecule, while similar to the core structure, is not identical. Therefore, it is determined to be unprotected. In addition, we also provide the download function of molecules. The specific operation is select molecules, click the download icon, and choose the desired file format and parameters. Scene 4. Target validation and target discovery. SciMind can search for potential targets of diseases, potential indications of targets, and potential drugs for diseases. We will start with a practical application, exploring targets relevant to clear cell renal cancer. Clear cell renal cancer is a common type of kidney cancer. Through a simple query, SciMind can quickly obtain multiple targets related to it, such as PBRM1 and VHL. Next, let's take a deeper look at the target of PBRM1. SciMind provides detailed information, including its important role in kidney cancer and other diseases. For example, PBRM1 has also shown potential importance in melanoma. Finally, for specific needs, such as developing a drug that mainly reduces abdominal fat without affecting the breasts. SciMind can also provide targeted solutions. Through in-depth analysis, the assistant recommended targets such as PPARG and MC4R, because these targets play key roles in metabolic regulation, and the mRNA expression levels are significantly different between the abdomen and chest. During use, we discovered a surprising additional feature. As the model accumulates more knowledge, SciMind can now identify the tools and parameters used to create images. Upload a picture. Send to assistant. The assistant will quickly identify the production tools and parameters of the image. Test the other two pictures. After that, we can retrieve similar pictures by keywords and easily find tutorials on related tools, which is impossible to do using Google Similarity Search or GPT-4. This means you no longer need to feel curious or envious about someone else's beautiful pictures. With just a simple query, we can provide the necessary design references to make your images equally striking.